everyone, my name is Vivian Turner and I'm a research scientist at the Savannah River National Laboratory, and today I'm going to be discussing some recent work we've been doing on an in-house 3D aqueous phase and transport model. The in-house model I'll be discussing today is called Algae. Algae is a 3D aqueous phase and transport model developed at the Savannah River National Laboratory in the late 80s, early 90s, initially to examine thermal plume transport on site. This is a finite difference model that solves the mass, momentum, and energy equations for sediment, particulate, and dissolved tracers. For algae, we define a particulate tracer as dissolved tracer that's been absorbed onto sediment. Algae can perform these simulations for a variety of predetermined lakes, rivers, coastal bays, and estuaries. We argue that the algae model is unique as it also considers complex atmospheric effects such as wind stress effects, sensible and latent heat transfer, shortwave and long-wave radiation transfer, and this includes the effect of clouds, while also including aqueous physical processes such as tidal effects and buoyancy forces. There's been a series of validation studies performed over the years, and this includes real-world events, such as an accidental train collision in Graniteville, South Carolina in the early 2000s, where chlorine gas was released and later deposited into nearby bodies of water. Another study looked at simulating flow rate of a creek as it enters a nearby river in Tennessee, and even fate and transport simulations of tritiated water from an accidental release at the Savannah River site in the early 1990s. And this release made its way from the Savannah River site into the Savannah River, which then flowed into the coastal Atlantic Ocean. You can see an example of this in the figures to your right, which show algae simulations of current speed and tritiated water concentrations. Recently, we've made a series of developments, such as a new backtracking capability, which can allow us to determine potential sources of a pollutant should the material be detected, but a source cannot be immediately found. This capability does need further development. However, we've found some success using a Bayesian approach, and we're able to narrow down the source to an 11-kilometer radius. Other recent work has added to the potential capabilities and applications for algae, and this includes a pilot study to be able to simulate microplastics, in addition to be able to simulate the physical process of coagulation and breakup of sediment, and then finally the ability to use runoff to understand urban heat island effects due to urbanization. Starting with the ability to simulate microplastics in algae, we wanted to make the argument that microplastics can serve as a potential carrier of radionuclides in the environment. And this is because, one, it is generally known that microplastics are everywhere in the environment, can travel long distances, and have even been found in remote environments. And two, there have been a series of lab studies performed that examine microplastic interactions with various radionuclides. So because of this, we want to know if we can simulate the movement of microplastics in algae and the absorption of radionuclides onto microplastics. To simulate microplastics in algae, we substituted the sediment properties in algae with microplastic properties. In this case, we replaced sediment density and diameter with microplastic density and diameter. We also utilized absorption coefficients, desorption coefficients, and decay constants of various radionuclides, which you can see in the table here from the literature. For this study, some of our absorption coefficients are actually based on radionuclides absorbed onto sediment. So for some of these runs, we are assuming that the absorption rate for the radionuclide absorption onto sediment is the same as a radionuclide absorbing onto microplastics. All microplastics for this study were also assumed to be the same type, and in this case, we chose polyethylene. Overall, we saw some success in our preliminary results, which you can see with time series plots for a dissolved release of cesium-137 at the top and bottom. For the top, we have no desorption terms considered, and the bottom time series plot considers both absorption and desorption terms. Overall, there are still some gaps in understanding the exact chemical interaction that occurs between microplastics and radionuclides of interest, which we have a colleague that is doing some experimental work on that now. So we're hoping that once we better understand this interaction, we can work towards incorporating that chemical reaction into algae. Earlier this year, we published work on the addition of sediment coagulation and breakup in algae. This allows us to create a more realistic environment that materials of interest can travel in. So if we can account for how suspended sediment can coagulate or break apart as it flows downstream, we can better understand how the subsequent material may either adhere to the sediment and flow farther downstream, or adhere to the sediment that coagulates with other sediment and then sinks to the bed floor. To do this, we performed a sensitivity study focused on clay sediments to answer the following questions. What is the behavior of suspended clay coagulation and breakup in a freshwater environment produced by algae, and how does this compare with the existing literature? How do these results change with a salinity-driven environment? And does the addition of turbulence through Reynolds number affect these results and lead to a significant change in flock size distribution and overall transport? To answer these questions, we broke the process up into 3 steps. The first was to create an addition of coagulation and breakup terms into the model's source code. The second was to develop and run a series of simulations, and the third was to perform a statistical analysis on the results to determine if there's any significant changes that occur between the simulations. To create the addition of coagulation and breakup into algae, we first divided the sediment and particulate contaminant into 3 bins or 3 size ranges. We then determined the fraction of mass for each bin. And once the fraction of mass is determined, we performed a bilinear interpolation based on a range of Reynolds numbers and salinity values. By performing this bilinear interpolation of the fraction of mass for each bin, we can then make sediment and particulate concentrations a function of salinity and turbulence, which are 2 major processes that control coagulation and breakup of sediment. We performed a sensitivity study based on 28 simulations of a hypothetical release at a facility with various sediment diameter densities and bin fractions as shown in the table above. Results include a time series and t-test for 12 locations along the Chesapeake Bay, shown in the figure on the right, to determine if significant differences are present both spatially and temporally due to changing these parameters. In this case, we chose the Chesapeake Bay, which is located on the east coast of the United States around the Washington, D.C. area, as overall this is a well-studied body of water, and due to large amounts of sediment input that goes into the bay every year, there's a large amount of data and literature available for us to compare results to. To show a subset of the results from our simulations, we have time series results for all 3 bin sizes at the top and t-test results at the bottom. These results consider both salinity and turbulence. The left-hand side of figures show results for sediment concentrations, and the right-hand side shows results for particulate concentrations. For the time series plots, you can see results for all 12 locations for sediment concentrations, whereas for particulate, we only have the source and locations 1 through 5 plotted because the particulate tracer only traveled to the middle of the bay after the 48-hour period. Overall concentrations of sediment produced similar results to the literature, and concentrations reached equilibrium fairly quickly. Particulate concentration shows peak values that occur roughly around the half-day mark for locations 1 and 2, and peak values comprise mostly of bin 3 sediment, which is our largest density and diameter. The t-test results below show consistent p-values across locations when salinity and turbulence is considered, which indicates to us a more predictable environment compared to runs where only salinity is considered. We also see that for particulate tracers, significant differences only occur at the locations closest to the source of release. The third application that we tested in algae is the ability to examine thermal surges due to runoff from storm events in a city where there's urbanization. To do this, we examined potential hydrological urban heat island effects of the Savannah River as it flows through the downtown Augusta, Georgia metropolitan region using HEC-HMS to estimate runoff, skin temperature, and algae to determine the subsequent fate and transport of the thermal plume downstream. We first performed HEC-HMS simulations, which utilized MRMS rainfall data, stream gauge data, and atmospheric data during a rainfall event, such as temperature and pressure, to estimate runoff in the Augusta metropolitan area. Algae then used the runoff estimates produced by HEC-HMS and then the skin temperature as input into algae. Algae was able to simulate temperature fairly close to what was measured from the stream gauge data. However, the temperature surge that we're looking for, which you can see in this bottom plot in the light blue, or the sudden increase in temperature by roughly 1 degree Celsius, was not produced by algae. And we think this may be a result to how runoff was estimated in HEC-HMS. So future work would involve a further investigation into runoff prediction and if similar results are produced when examining potential runoff of, say, particulate tracers. In summary, we've completed recent work to develop new capabilities and applications for algae. This includes accounting for microplastics, adding the physical process of coagulation and breakup of sediments, and examining the potential for simulating aqueous urban heat island effects. There's a lot of opportunities for future work, and this includes incorporating other models, such as atmospheric models to account for deposition into bodies of water, or oceanic and wave models to account for more global fate and transport. Incorporating experimental data will also allow us to better account for interactions for radionuclides and microplastics. And currently, the algae model can run in a clustered GPU environment or an app-based environment, and the app-based environment could also use some technical improvements. I want to thank you all for taking the time to listen about algae, and if you have any further questions or would like to discuss potential collaboration opportunities using our algae model, I've listed my email here for you all to reach out.